Hello everyone, my name is Lizet Garcia and welcome to my presentation on the effects of feeding rumen-protected lysine during the transition period on postpartum immunometabolic gene expression of Holstein cows. For the past year, I had the opportunity to work at our school's dairy cattle research unit, where the Dairy Focus Lab was conducting research to learn more about the impact amino acids may have on the transition period of a cow. So what is the transition period? And why is there much attention to this stage? Well, when a cow transitions from the gestation period to parturition, that is what we consider the transition period. It's usually 3 weeks before to 3 weeks after calving. According to Dr. James Drakely from our Department of Animal Sciences, the transition period is incredibly difficult to manage because cows are reducing their feed intake and increasing their chances for developing metabolic diseases. Although there have been trials examining the effects amino acids have on milk production, not much data has been shown about the influence amino acids have on gene expression profiles of the endometrium. Therefore, we hypothesize supplementing dairy diets with rumen-protected lysine may improve the reproductive immune response and metabolism. The trial consisted of a total of 73 multiparous pregnant Holstein cows that were 305 days mature equivalent milk production. They were randomly divided to 4 dietary treatment groups. LL groups were fed rumen-protected lysine pre- and postpartum. LC were only fed rumen-protected lysine prepartum and not postpartum. CL were not fed rumen-protected lysine prepartum but rather postpartum. And CC were not fed any type of rumen-protected lysine. Treatments began 28 days before expected calving date and were fed until 28 days relative to calving. Following the 28 days relative to calving, endometrial tissue samples were retrieved from cows who met specific health qualifications, such as having a Matricex and smell score less than 3, and normal ranges of body temperature, pulse, and respiration. The samples were then tested for genes related to inflammation and metabolic protein biomarkers using mixed procedures in SAS. In Figure 1, you can see the anatomy of a bovine uterus, and the samples were collected from the uterine body via biopsy. Once we extracted the RNA from the endometrial tissues, we evaluated a variety of genes related to the immune response and metabolism, which can be seen in Table 1. However, we only noticed an effect for mucin 1 mRNA, mucin 4 mRNA, and interleukin 10. In Table 1, you can see there was a tendency for an effect of cow's L postpartum with a p-value of 0.06 for both mucin 1 and mucin 4 mRNA. Figures 2 and 3 show there is a greater gene expression of mucin 1 and mucin 4, and they are highlighted with an asterisk next to them. These indicate that there seems to be more of an effect on cows fed lysine after calving. Both mRNAs serve a role in protecting the reproductive tract mucosa from bacterial invasions by lubricating the reproductive lining as a protective barrier. We also observed cows fed lysine both pre- and postpartum had greater expression of interleukin 10 than any other group, which can be seen in Figure 4. Interleukin 10 has a beneficial function by increasing antibody production to fight the risk of cows developing a reproductive tract inflammation disease. Overall, these results indicate that feeding rumen-protected lysine postpartum does enhance the uterine immune response due to an increased expression of mRNAs and cytokine. Both mucin 1 and mucin 4 allow for a greater defense mechanism against bacterial infections, while interleukin 10 increases anti-inflammatory responses. All of this can improve the fertility of cows and ultimately increase the efficiency of dairy production. I would just like to end by saying thank you to the Dairy Focus team and the staff of the Office of Undergraduate Research for allowing this opportunity to happen.